Hey there, how you doing? Flipped Chemistry. This is lecture number 31. We're almost done. We are continuing our conversation of organic chemistry today. We're looking at biomolecules. This lecture is entirely review from biology. You had all of this before. So hopefully this is an opportunity for you to remember as opposed to learn, okay? Here we go. Biochemicals, the chemicals of life, are called polymers. All of them are. They're all big, long, huge molecules, and they're made up of smaller repeating units called monomers. And so in all of the examples we're going to show you today, there are smaller molecules, not all of them small to begin with, but smaller molecules that can be strung together to make very large molecules, okay? That process is called polymerization. I'll show you an example of that in a minute. The first of the four biomolecules are called carbohydrates. Carbohydrates are sugars, and they are used by living things for energy. All living things use sugar for energy, and simple sugars are called monosaccharides, one sugar molecule sugars. Simple sugars, they're the monomers, and they're called monosaccharides. These are used by all living things for short-term energy, quick energy bursts. When you eat food that has a lot of sugar in it, that goes straight into your bloodstream, and your cells can jump on the production of energy immediately, okay? Here's a picture of polymerization real quick before we move on with carbohydrates. Polymerization happens when a molecule's alcohol group and another molecule's hydrogen get kicked out, and that makes water. And so then those two molecules form together. Here's another alcohol group that's going to leave, and a hydrogen, and that forms water, and then those bonding sites that are exposed form a polymer. So polymerization is sometimes called dehydration synthesis because you're always removing these little water molecules to make these things grow. Alcohol groups tend to be the place where biomolecules stick together. Okay, sugar, simple sugar, glucose, fructose. Both of these are simple sugars. They're monosaccharides. They're one sugar long, right? Monosaccharide, one sugar. The only difference between them is where this oxygen is bound. Is it bound to a terminal carbon, or is it bound right here? So this oxygen bound to the next to the last carbon makes a carbon and an alcohol group hang out there. The oxygen bound to the last carbon makes just the alcohol group hang out there. Your body processes them almost identically. This is the sugar that we get from like beets and cane. This is the sugar you get from like strawberries and bananas. Okay. Now, when you polymerize a carbohydrate, you can make two different kinds of things. You can make what's called a complex carbohydrate, or a complex carb, and that is just a sticking together of two sugar molecules into something called a disaccharide. Disaccharides take just a moment longer for your body to get the energy out of. So if you eat something that has mono and disaccharides in the meal, your body will go after the monosaccharides first, give you a quick burst of energy, and then it'll go to work on breaking the disaccharides into two molecules so that it can go through the process of breaking those down and getting energy out of it as well. Disaccharides, two sugar molecules. Sucrose is an example of that. And then there are polysaccharides that have not just three, but like thousands, tens of thousands, hundreds of thousands of sugar molecules all strung into a fiber. And there are two ways of doing that. Plants tend to be the ones that make really complex carbohydrates like polysaccharides. Animals almost never make polysaccharides. If we need to store energy long term, we use fat. But a plant, if it needs to store energy long term, will make starch. And that is a chain of hundreds of sugar molecules. And the starch tends to be stored in certain parts of the plant, and that's where we get potatoes and yams and all kinds of tubers from, right? Starches are just chains of lots of sugar molecules for energy storage. And then there's another way of chaining a bunch of sugars together, and this one is not for energy storage. This is for structure. The plant is built out of a material called cellulose. Cellulose is not a protein. Cellulose is a massive, massive polysaccharide. And it is strings of hundreds of thousands of sugar molecules in a row. And these fibers of sugar molecules get wound up and woven into the body of the plant. Okay, so just some pictures real quick. These are disaccharides: sucrose, lactose, and maltose. This tends to be in grains, this tends to be in milk, and this tends to be in other vegetable sorts of things. Okay, fruit sorts of things as well. Sucrose, two sugar molecules joined here this way, a unique bond. If you look, basically the bond is what's different between all of these, right? The way that the geometry of the bond is arranged. So these are disaccharides, two sugar molecules stuck together. This is cellulose. Chains of hundreds of thousands of sugar molecules make like threads. Those threads get wound around each other into something called a microfibril, and those microfibrils get woven like a carpet into a very thick, very strong material called cellulose. That is what the plant makes its cell walls out of. How strong is cellulose? Well, wood that your house is probably built out of is cellulose. So pretty darn strong. And it's sugar. Lipids are the next kind of biomolecule. Lipids are more the domain of animals, whereas plants are kind of the kings of carbohydrates. Animals are kind of the kings of lipids. Now, it's not that plants don't make lipids, but they don't make them like animals do. The monomer of a lipid is already a fairly large molecule called a fatty acid, and it's several carbons long, 20, 30, 40 carbons long sometimes. And these fatty acids, they end in a carboxylic acid group, which is why they're called fatty acids. And they are the basic unit of fat. When your body has extra energy, it might polymerize some saccharides into some disaccharides for a minute and store it in the liver and see, do I need this in the next 20 minutes or an hour? But if it's not needed after that point, it takes those sugar molecules and it does a little chemistry on them, and it turns them into fatty acids. And fatty acids are the long-term energy storage in animals. They are very nonpolar. They do not mix with water. And that allows them to be good for cell membranes because water does not readily pass across this. You can have little vacuoles, little bubbles made up of fat, and the water is pretty much trapped in that bubble of fat. The polymer of a fat is called a triglyceride. It's three of those fatty acids joined by a molecule called glycerin across the top. And the glycerin attaches to all three of the carboxylic acid units, and so we have three fatty acids on a glycerin molecule, and we call that a triglyceride. Okay? Now, saturated and unsaturated. If something has every hydrogen that it can have, we call that saturated. So every carbon here has two hydrogens. It is fully loaded. Can't have any more hydrogens than that. This is a saturated fatty acid. This is all single bonds. A unsaturated fatty acid has at least one double bond or maybe even a triple bond. And what that does is it means that this carbon only has one hydrogen, and so does this one. So these are not saturated. You could, in theory, add more hydrogen to this molecule if you were to take this double bond and make it a single bond. Unsaturated fatty acids are a lot better for your body because when your body goes to break down fat and says, okay, I'm ready to use that energy, it starts the chemistry at the double bond, which means it's going to break this apart and then digest this way and this way. And this is a lot easier to get started. Over here, the chemistry is looking for a double bond to start with, and the only thing it's got is over here, and it's a lot harder. So your body tends to store saturated fat for later, and when it really needs energy, and it has to work hard enough to digest these harder to digest molecules. So that's why unsaturated fatty acids tend to be good for your body, and saturated fatty acids tend to be bad for your body. Okay, as you saw from the picture before, saturated fatty acids are straight chains. And unsaturated fatty acids kink, they bend. So this is a picture of an unsaturated triglyceride because it bends and it bends and it bends and it bends. And all of these bends you see are places of unsaturation. So this would be a polyunsaturated fatty acid molecule, triglyceride. And this would be something that your body could digest when it's ready to use that energy. This one is even more kinky, more twisted. And this would be even more of a polyunsaturated triglyceride. Okay, steroids are also fatty acids, but they're fatty acids that instead of having three long tubes stuck to one triglyceride, it's a fatty acid molecule that has been folded and wrapped and attached to itself to make a series of rings. And those rings are used as chemical messengers. So when the body wants to send a message from one place to another, it can do it electrically through your nerves. But if it's not your nervous system specifically sending the message, then it's usually done with a hormone. And a steroid is a kind of hormone. And this hormone is secreted by one part of the body, floats through the blood, and the chemical message lands somewhere else. And the body says, oh, I'm supposed to start doing this. Steroids, hormones, are fatty acids, but they're fatty acids that have been like crocheted into loops. So it's not so long a molecule anymore because it's been turned into a bunch of loops. But this would be cortisone. This is cholesterol. That's a bad steroid. This is vitamin D. This is testosterone. So all of these are steroids that are fat molecules that have been woven into a unique shape that bears a message around the body. Proteins are amino acids. Amino acids are the monomer. We said that at the end of last lecture. And when you join amino acids together, the kind of bond that you form is called a peptide bond. Remember, all of this is dehydration synthesis. Water leaves, but the particular kind of bond there is called a peptide bond. So when you string lots of amino acids together, you have polypeptide bonds. You have more than one peptide bond, and that's why sometimes proteins are called polypeptides. But proteins are strings of amino acids. Now, this is one of the most amazing examples of intelligent design out there. The information for you is in DNA, and we'll get to that in the next biomolecule. But that information is read, and it's turned into a string of amino acids that are all joined together in the right order to make a protein. That's one level of information. But that string of amino acids is not done. It's not ready to do its job. It has to be twisted and woven and folded on top of itself to make some kind of three-dimensional shape. That's another level of information. And then sometimes there's a second kind of folding, that once it's made this kind of folding, now it folds again. And that is a third level of information. And then oftentimes these proteins have to work with other proteins that have also been folded two different kinds of ways, and they have to work together and fit together like pieces of machinery. That's a fourth level of information. The level of information that it takes to make DNA create amino acids in the right order already is astoundingly complicated. And then when you ask that to wrap up and spin and then fold again and then work with something else that has done that, you're multiplying all these probabilities of complexity. The degrees of complexity get multiplied. And it is an astoundingly high degree of complexity that represents any functional protein unit. To look at this and not believe in a God is absolute folly. It absolutely is. There is no way that this high degree of complexity can randomly evolve. Saying it's like having the encyclopedia come out of a printing press when you drop a bomb on it is not even close. The degree of complexity of the Encyclopedia Britannica comes nowhere near the degree of complexity of a functional protein. So it is absolutely absurd, it is ludicrous, it is foolish to look at a protein and say, oh yeah, that just happened. But you know, the Bible tells us that the fool says in his heart there is no God. So let's look at some examples of this. Here we have amino acids in the right order. And this is done by the cell. It's actually DNA that does the work to read DNA, to put proteins in there. So, I mean, there's more and more levels of complexity as we go here. But let's say you get these amino acids in the right order. This string of amino acids is not all that there is to this protein. It has to get folded, and sometimes it gets wound up in a spiral. And the folding, like this spiral, then gets folded again. So spirals are not enough. We have to have spirals folded around on itself to make stacks of things. And that's the tertiary structure. And then this protein would work with, in this particular picture, 3 other proteins to make a quaternary structure, to make multiple proteins functioning together to do one job. This is insane amount of organization and complexity, and this screams the need for a designer. Okay, the fourth kind of biomolecule is a nucleic acid. Nucleic acids are where the information for all of that comes from in the first place. Genetic information is coded in a language that has 4 letters. Those 4 letters are called nucleotides. Those are the monomers of nucleic acids. The polymers are either DNA or RNA, and we string them together, and so a whole bunch of amino acids makes a piece of DNA or a piece of RNA, depending on what sort of monomers you have and the purpose that it is being made for. A segment of DNA that codes for a particular trait, that says this is how you do this, we call that a gene. Genes can be thousands, millions of base pairs long. And then several genes worth of DNA or RNA will be lumped together into a unit called a chromosome. A chromosome is phenomenally complex, and you have 23 pairs of these chromosomes in every single cell of your body. To say that there's a lot of DNA in you is like saying water is wet. It doesn't even get to the truth of the matter. Every cell of your body has about 2 meters of DNA in it, and you are something on the neighborhood of 12 trillion cells. It's 12 trillion times about 2 meters if you were to multiply that out, the DNA in your body does not just reach to the moon and back. The DNA in your body does not just reach to the sun and back. The DNA in your body will make dozens of laps between the Earth and the sun. It's astounding how much DNA is in you. And again, this is all evidence of design. So most organisms have lots of chromosomes. This is a picture of an amino acid. It has a 5-member sugar and a phosphate group, and then whatever the amino acid base group is. This is where the information is. This is the data. A whole bunch of these amino acids string together. A bunch of these nucleotides string together, and the order of the nucleotides is the way that it's read. We went through this in detail in biology. This is just review. On one side of a DNA molecule and on another, the opposite anticodons will pair up. So cytosine always pairs with guanine, adenine always pairs with thymine, and they tend to be very easily reproducible. Given one side, you can make the other. And then that whole structure is wound into a double helix to protect the information on the inside, and we have DNA. Just so you know, I'm not the crazy one. Francis Collins, the head of the Human Genome Project, started understanding DNA as the head of the Human Genome Project as an atheist or an agnostic, and he finished as a Bible-believing Christian, because the amount of information and complexity in here is something that cannot happen on its own. So praise the Lord for his creation. If you have any questions at all, you can put them in the comments field below, and I will get to them as soon as I can, or I will see you tomorrow in class. Jesus loves you, and so do I.